We're going to be talking about phage genomics 2, which is taking phages from last semester that were identified and then sending them in sequence and analyzing their genomes. So this is actually a picture of a phage infecting a bacteria, and those letters there represent an amino acid sequence from one of our genes. So what are we actually doing? We're taking, and why are we looking at their genomes and analyzing them? So we're taking and going through all of the genomes to try and identify functions for each of the genes and confirm where their position is in the genome, as well as try and identify which clusters and subclusters that phages will fall into. And all the colors on this wonderful little graph here are different clusters. So all the phages in that cluster will have 50% genome homology. So the tools that we use to do this for every gene is DNA master, Famrator, coding potential maps, StarDater, BLAST-P, and HHPRED. So DNA master is our home base. We took our novel phages, sent them off to be sequenced, and their entire genome was then uploaded into this program. And so in the blue little box there, we're looking at, in this case, gene 43, and it gives us our 5' and 3' ends as long as how long our gene is. And then in the red box over here are all of our annotation nodes. So where our gene actually starts and stops, and all the information that we gathered from it. In the purple box up here is a pictorial representation of all the different genes in the genome, and by selecting one of those, we can then, in the green box down there, look at the ribosome binding site scores, and depending on which start for the gene we use the best ribosome binding site that we can. So Famrator is the next thing that we do, which is just a pictorial representation of all that awful words and numbers that you saw on the previous slide. And so the tape measure underneath is actually our phage genome. And each box represents a different gene, with the number in the box corresponding to that gene's position in its genome. And the colors of the box, when they're the same colors, they have the same function and are in the same family. And we can use that to figure out what our gene actually does. So coding potential maps and StarDater are two more of the ways that we try to confirm our starts and stops. And on the right, for Climon gene 15, that's an ideal coding potential. So every single one of the codons in that black bar is actually coding for something and active. While gene 17 for Climon is also a very good coding potential map and would be more than sufficient. StarDater will then give us different possible starts that the gene might have. So if for some reason this first part of this gene had no coding potential, we could then change the start to give it the best ribosome binding site score that we can. Once we're done with that, we'll take the amino acid sequence from DNA master and plug it into what's called BLAST-P, which takes all of the other sequence phages that have existed and compares our gene of interest to that gene looking for homology. And the query at the top is our gene of interest, and each colored line below is a possible hit or a possible match that shows how much of the gene it actually agrees with. So this is then what we get out of BLAST-P, and the biggest things that we're looking for to determine whether this is a good match or not is in the green box, the most important being query cover and e-value, and the query cover is just the extent that the gene is matched, that the possible function of gene matches to our gene of interest. And then the e-value is the probability that this match for a function could happen by chance. So 0.0, as we have in the blue box, is an ideal e-value score. HHPRED does a very similar thing. It looks for domains that are consistent in all other sequenced organisms. So it'll just look for even the smallest homolog in any part of a gene to try and figure out its function.
So over this semester, also. Yeah. So over this semester, we have successfully annotated 100 genes of time-bond, which is over 68,000 BTs length. It's so long that even one single slide cannot fit that many genes. We have to use 2 slides. Yeah. So we've also annotated 88 genes of Gantro, which is over 53,000 BTs length, and it's also long. So I want to say that there's really great work done by our lab in this semester. I want to thanks to my peers and my professor, Dr. Gao. Yeah. Yeah, so now we have many bioinformatic tools, as Kay has said, but sometimes they can't make the calls. For example, in this DNA master display of gene 28, you see the red box there. That is two different kinds of bioinformatic tools. The glimmer and the gene mark cause a different start. So as the annotator, we have to collect as much evidence as we can, and based on all this evidence, we would finally determine the start and the function of the genes.
Alright, so if it comes back, we have more examples of why you need it. Oh, fantastic. So what 2 of our classmates, Jade and Sophie, did while going through Climon was realize that we need to delete and add genes, and found where that needed to happen. So gene 9 in that green box was decided to be deleted because, as you can see, none of the other B1 cluster phages have that gene, and it's also extremely small, so it's not likely, and had no function when blasted, so it's not likely to actually be coding for anything. While that gap that's in the light blue box was then added to the genome because all the other B1 cluster phages called that gene as an actual gene and not just an amino acid sequence gap. Sorry, gene code. So then what we did was we took the nucleotide sequence and converted it to an amino acid sequence and blasted it and got a function for it. And so in the green box at the top, it shows that it is also called in other B1 cluster phages, and with a good e-value and good query cover. And then the blue box lower down actually shows a possible function for the gene. So after taking all that information into consideration, it was decided that we would add that gene, which DNA Master and all the other programs didn't just do automatically.
So now, may I present you tape measure protein? I want to ask a question. In this family map, among all those genes, which one of these is the longest? The answer is obvious. It's gene 28, as you can see. And it is tape measure protein. Since it is longest and can be found in every phage, so it is easiest to annotate. But why are we looking at tape measure protein? Tape measure protein assembles the tail, as shown in this picture. Those 2 yellow dots represent the length of the tape measure protein, and as you can see in the right-hand side of this picture, the length of the tape measure protein will finally determine the length of the tail, as like that. Here's another interesting region. This is the family map of GP70 to GP80 of time-bond. According to our annotation, GP73 is a DNA binding domain, and GP75 is a translation, initiation, and elongation factor. But GP71 is called PLA2 line domain, which is different from all the other transcription and translation regulators. So, and what's more interesting is through HHPreg, we found this GP71 PLA2 line domain in all B1 cluster phages. This inspired us to go on our research. Nonetheless, not only this GP71, but also the entire GP70 to GP75 region was found in all B1 cluster phages. As shown in this, those Kleinborn, Menard, and Olive is all B1 cluster phages, and they all contain this region. This might contribute to the evolution of all the B1 cluster phages. So we would like to do some related research about that. But we are bioinformatics classes, so we can't do that much. So for our future direction, we will go back from those bioinformatic tools to the bench and knock out this region and try to see which kind of effects it would have on phages. And if there's really some significant effects, it could be used in phage therapy as the previous lab has talked about that. And here's our references. Yeah. We would like to thanks to our professor, Dr. Gao, and MCDD Department of Steel Boulder, and all the other sponsors, as the professor has talked about.
Can we answer any questions anyone might have? Because it's taken us a semester to figure this out, and we're still not even entirely sure. So I'm sure you guys have something. Yeah. Oh, yeah, Fred. Gotta love Fred. It's highly inaccurate and will never give us something that we want because it actually compares everything to, compares our gene to all sequenced organisms instead of just phages and phage hosts. So, it's great. All right, another question? No, I thought there was another one. Anybody else?